For a long time, we thought most of our DNA was useless, and when I say most, I mean almost all of it. The number floated around was 98%. Imagine buying a massive 1,000-page book. You open it, excited to read the story, but you find that only 20 pages have words on them. The other 980 pages are just blank or filled with what looks like random gibberish. That's what looking at the human genome felt like for a while. Scientists found the parts that mattered, the genes. These are the instructions for building proteins. Proteins are the little machines that do everything in your body. They digest your food, carry oxygen in your blood, fight off viruses, and build your muscles. Genes are the blueprints for these machines. They make up about 2% of our genome. So, what about the other 98%? For decades, the honest answer was, we don't really know. So it got a nickname, junk DNA. It was a label born out of confusion, not confidence. It wasn't a formal declaration that this DNA was useless, it was more of a shrug. A placeholder for a mystery we hadn't solved yet. It seemed repetitive, it seemed random. It didn't seem to do anything. The central idea in genetics was pretty simple. DNA makes RNA, and RNA makes protein. This was the clean, elegant assembly line of life. If a stretch of DNA didn't end up as a protein, it was seen as being off the assembly line. It was just not there. Maybe it was evolutionary leftovers, scraps from old projects, fossilized bits of genetic code that our ancestors used but we no longer needed. Like a digital attic full of old, forgotten files. But science is a story of mysteries being solved. And the story of junk DNA is one of the biggest turnarounds in modern biology. We were wrong. It's not junk, not even close. That massive 980-page section of the book we thought was gibberish, it turns out it's the instruction manual, it's the table of contents, the index, the editor's notes. It's the conductor of the whole genetic orchestra. Without it, the 20 pages of music would be a chaotic mess. The first hints that something was up came from looking at other animals. Scientists would compare the genome of a human to a mouse or a fish. They found something strange. A lot of this junk DNA was the same across species. It was conserved. Evolution is ruthless with things that are useless. If a piece of DNA truly does nothing, mutations can accumulate in it without any consequence. Over millions of years, it should become completely different between, say, a person and a dolphin. But large chunks of this non-coding DNA were almost identical. Why would evolution carefully preserve junk? It wouldn't. If something is being kept around for hundreds of millions of years, it's because it has a job. A very important job. Deleting it would be a bad idea. This was the first major crack in the junk DNA theory. This junk had to be doing something. We just didn't know what. The big breakthrough came when we stopped thinking about DNA as just a list of parts. We started thinking about it as a system. A computer doesn't just need software files. It needs an operating system to tell it when to open which file, how much memory to use, and how to make different programs work together. The junk DNA is our genome's operating system. A huge part of this operating system is about regulation. It's about control. Think about your body. You have a liver cell and a brain cell. They both have the exact same set of genes, the same DNA blueprint, but a liver cell acts like a liver cell and a brain cell acts like a brain cell. They look different and do completely different jobs. Why? The difference is which genes are turned on and which are turned off. A liver cell turns on the genes for detoxification. A brain cell turns on the genes for sending electrical signals. They use the same cookbook, but they're reading from different recipes. What tells them which recipe to read? The junk DNA. Large stretches of this non-coding DNA are filled with switches. These are called regulatory elements. There are promoters, which are like the on button for a gene. They sit right next to the gene and signals start reading here. But it gets way more complicated than that. There are also enhancers. An enhancer is like a volume knob. It can be very far away from the gene it controls, thousands or even millions of letters of DNA away. But the DNA molecule is a long tangled string inside the cell's nucleus. It can loop and fold back on itself. An enhancer that's far away on the linear sequence can, through this folding, end up physically right next to the gene it needs to regulate. When a specific protein called a transcription factor latches onto this enhancer, it cranks up the activity of that gene. It tells the gene not just to turn on, but to turn on a lot. Then you have silencers. They do the opposite. They're like a mute button. When the right proteins bind to them, they shut a gene down or at least turn its volume way down. There are also insulators, which are like little fences. They stop an enhancer for one gene from accidentally turning on the gene next door. They create boundaries and organize the genome into neighborhoods. So, this massive sea of junk is actually a complex control grid. A landscape of switches, dials, and fences that choreograph a beautiful dance of gene activity. This is what allows a single fertilized egg with one set of DNA to develop into a person with hundreds of different cell types, all working together. Every cell uses these regulatory regions to create its unique identity. It's not about having the genes, it's about how you use them. And the junk DNA is the user manual. This discovery has changed how we think about disease. For a long time, when scientists were looking for the genetic causes of diseases like diabetes, heart disease, or schizophrenia, they focused on the genes themselves. They looked for mutations in the protein coding 2%, sometimes they found them. But often they came up empty. Then, a new kind of study became common called a genome-wide association study, or GWAS. These studies scan the entire genomes of thousands of people, some with a disease and some without. They look for tiny differences in the DNA letters that are more common in the people with the disease. The results were stunning. Over 90% of the genetic variations linked to common diseases were not in the genes themselves. They were in the junk DNA. Suddenly, it all made sense. A disease might not be caused by a broken protein. It might be caused by a broken switch. A mutation in an enhancer might mean a crucial gene is never turned on enough. Or it's turned on too much. Or it's turned on in the wrong cell at the wrong time. A heart disease gene might be perfectly fine, but the enhancer that's supposed to turn it on in developing heart cells has a typo. The result is a malformed heart. An autoimmune disease might be caused by a broken silencer that fails to keep a self-attacking gene turned off. The complexity is mind-boggling. A single gene might be controlled by a dozen different enhancers. One enhancer might turn it on in the liver, another in the kidney, and a third in the skin during the third week of embryonic development. A single mutation in just one of these enhancers could have very specific consequences. This is the new frontier of medicine understanding the grammar and syntax of our regulatory code. But the story doesn't end with regulation. There's another huge piece of the puzzle. It turns out that a lot of our junk DNA isn't silent at all. It gets read by the cell. It gets transcribed into RNA just like a regular gene, but these RNA molecules never go on to become proteins. They are the final products. For a long time, these were also dismissed. What's the point of making an RNA blueprint if you never build the machine? This is the world of non-coding RNA, or ncRNA, and it's another massive function of the junk DNA. We're discovering an entire new layer of biology run by these molecules. They are not blueprints, they are tools and signals in their own right. There are many types. One famous group is called microRNAs. As their name suggests, they are tiny. Just a handful of genetic letters long, but they are incredibly powerful. A microRNA works by finding a regular protein-coding RNA message, the kind that is supposed to become a protein, and sticking to it. When it does that, it's like sticking a piece of tape over the barcode on a product at the supermarket. The cell's machinery can't read it properly anymore. The microRNA either blocks the RNA from being turned into a protein, or it marks the RNA for destruction. So microRNAs are another layer of control. Even if a gene is turned on and transcribed into RNA, a microRNA can come along and say, nope, we're not making this protein right now. It's a fine-tuning mechanism. It's estimated that microRNAs regulate more than half of all our genes. They're involved in everything from cell growth to brain function. When they go wrong, they are implicated in diseases like cancer, where cells grow out of control. Then there are the long non-coding RNAs, or lncRNAs. These are much bigger molecules, sometimes thousands of letters long. Their world is newer and more mysterious, but what we're learning is incredible. They seem to be the ultimate multitaskers. Some lncRNAs act as scaffolds. They can grab onto different proteins and bring them together in one place, creating a little functional hub. Imagine needing to assemble a chair. An lncRNA is like a jig that holds all the legs and the seat in the right position so you can screw them together easily. Other lncRNAs act as guides. They can bind to a protein and carry it to a specific spot on the DNA, telling it where to do its job. Some can even directly attach to the DNA and change its physical shape, making it easier or harder to read the genes in that region. They are a huge and diverse class of molecules, and we're just beginning to understand their language. What's clear is that this junk DNA is actively producing thousands of these functional RNAs that act as a hidden network of control and organization within our cells. There's even a more basic physical role for some of this DNA. It's not all about information and regulation. Some of it is purely structural. It's the nuts and bolts of our chromosomes. Think about your shoelaces. They have those little plastic tips on the ends called aglets. Their job is to keep the shoelace from fraying and unraveling. Your chromosomes have something very similar. At the very end of each chromosome is a long, repetitive stretch of DNA called a telomere. It doesn't code for any protein. Its sequence is just the same 6 letters repeated over and over, thousands of times. T-tag, T-tag, T-tag. This is junk, DNA in its most classic form, simple and repetitive. But its job is essential. Every time a cell divides, it has to copy all of its DNA. The copying machinery isn't perfect, and it can't quite copy the very, very end of the chromosome. So, with each cell division, a little piece of the end is lost. The telomeres act as a buffer. The cell loses a bit of the repetitive telomere sequence instead of losing an actual important gene. It's like a disposable cap. The length of your telomeres is even linked to aging. As they get shorter over many cell divisions, the cell eventually stops dividing, or dies. They are a countdown clock for the cell. Another structural piece is the centromere. This is a large, constricted region on a chromosome, also made of highly repetitive young DNA. When a cell divides, it needs to pull its copied chromosomes apart so that each new daughter cell gets a full set. The centromere is the handle. It's the spot where the cellular machinery grabs on to pull the chromosomes to opposite sides of the cell. Without this well-defined handle made of junk DNA, cell division would be a chaotic disaster, leading to cells with the wrong number of chromosomes, which is often lethal or a hallmark of cancer. So, we have the control panel, the non-coding RNA network, and the structural components. But there's one more part of the story of junk DNA, and it's arguably the weirdest. A huge chunk of our genome, maybe close to half of it, is made of what are essentially ancient invaders. It's the legacy of jumping genes and long-dead viruses. Let's start with the jumping genes. These are properly called transposons. They were discovered by a brilliant scientist named Barbara McClintock back in the 1940s and 50s. She was studying the genetics of corn kernels and noticed that their colors seemed to change in weird, unpredictable patterns. She figured out that there were bits of DNA that could literally cut themselves out of one spot in the genome and paste themselves into another. For this work, which was so far ahead of its time that many people didn't believe her, she eventually won a Nobel Prize. Our genome is absolutely littered with these transposons. Most of them are now inactive. They have accumulated mutations over millions of years and have lost their ability to jump. They are fossils, but their presence has had a profound impact. When they jump, they can land right in the middle of a gene, breaking it. That's usually bad, but sometimes they can land near a gene and act as a new switch, changing how that gene is regulated. Sometimes they carry a piece of another gene with them, creating a new hybrid gene. Transposons are a powerful engine of evolution. They shuffle the genetic deck. They create new possibilities. While many of their jumps are harmful, over a long enough timescale, a few of those jumps are beneficial, creating new traits for natural selection to work with. Our genome is a graveyard and also a playground for these jumping genes. They are a major reason why our junk DNA looks so strange and repetitive. We are looking at the accumulated remnants of millions of years of their activity. And then there are the viruses, specifically a type of virus called a retrovirus. HIV is a modern example. These viruses work by inserting their own genetic code into the DNA of the cells they infect. Sometimes a retrovirus infects a sperm or an egg cell. If that cell goes on to create an organism, the viral DNA will be in every cell of that organism's body. It will be passed down to its children and its children's children. It becomes a permanent part of that species' genome. These are called endogenous retroviruses, or ERVs. Our genome contains tens of thousands of them. Like transposons, most of them are broken and inactive. They are molecular fossils of ancient plagues that our ancestors survived. For a long time, they were seen as the ultimate example of junk. What could be more useless than a broken, dead virus stuck in your DNA? But again, we were wrong. Evolution is the ultimate recycler. It doesn't like to let things go to waste. Over millions of years, our cells have co-opted some of these ancient viral genes for our own purposes. The most stunning example is a protein called syncytin. This protein is absolutely essential for the formation of the placenta, the organ that connects a developing fetus to its mother. Without syncytin, the pregnancy would fail, and the gene that makes syncytin is not originally ours. It came from a retrovirus. The protein's original job in the virus was to help it fuse with a cell to infect it. Our bodies have repurposed that. Fuse together, function, to allow fetal cells to fuse together to form a critical layer of the placenta. An ancient enemy has become an indispensable partner in the creation of human life. And this isn't a one-off. We are finding more and more of these old viral bits that have been given new jobs, often as regulatory switches that control our own genes. So, when we look at the 98%, we are not looking at a barren wasteland. We are looking at a rich, complex, and dynamic ecosystem. It is a dense network of regulatory switches. It is the source of thousands of functional RNA molecules. It is the structural backbone of our chromosomes, and it is a living archaeological record of our deep evolutionary past, filled with tame jumping genes and repurposed viruses. The term junk DNA is now mostly dead among scientists, or used ironically. People prefer terms like non-coding DNA or talk about the dark matter of the genome, acknowledging how much we still have left to learn. The journey from junk to essential is a perfect example of how science works. It's a story of humility. We labeled something as junk because we didn't understand it. Our ignorance, not the DNA, was the problem. As our tools got better and our thinking expanded, the picture changed. What we've learned has huge implications. We understand that our complexity as humans doesn't come from having more genes than other creatures. A grape has more genes than a human. A simple water flea has more genes. Our complexity comes from the sophisticated way we regulate our genes. It comes from the depth and intricacy of our non-coding DNA. It's not about the number of instruments in the orchestra, it's about the quality of the music they play and the skill of the conductor. The conductor is the 98%. This new understanding is shaping the future of medicine. We're learning to read the regulatory code, to understand how typos in it lead to disease, and hopefully, one day, to fix them. We're exploring the world of non-coding RNAs as potential targets for new drugs. The junk that we once ignored has become one of the most exciting and important areas in all of human biology. It's a reminder that in the book of life, no page is truly blank. Sometimes you just need to learn how to read the invisible ink. And the story written there is more complex, more surprising, and more beautiful than we ever imagined. It's the story of how to build a human, and it's hidden in plain sight, in the parts we once threw away as junk.